everyone. Today I will be discussing the topic biology-driven biomaterials and minimally invasive dentistry, how it will forward men towards regenerative and conservative approaches. So we already know that the modern dentistry emphasizes the use of conservation and biological prevention rather than aggressive tissue removal. So we have to know about the biomaterials which have regenerative potential and are minimally invasive in technique, so that it can provide therapeutic interventions and the clinicians can in turn preserve the pulp vitality, ensuring long-term to survival. So what do we mean by biological prevention exactly? So instead of full aggressive caries excavation, selective caries removal is done, leaving affected dentine that can remineralize in future, preserving the pulp vitality. Biomaterials with regenerative potential specifically in use in the market today are biodentin and MTA, because they release calcium ions and stimulate the dentine bridge formation. In this, we incorporate the minimally invasive techniques such as the resin infiltration, that is for early proximal caries prevention, and ART, the atraumatic restorative techniques with high viscosity glass iron ore that conserve both the tissue and provides fluoride release. So coming to the other aspects of it, so why we have to have biological foundations first before ensuring the proper treatment? Because we have a complex pulp dentine system. The dentine pulpal stem cells, the shed differentiate into odontoblast-like cells, releasing dentine matrix proteins and forming reparative dentine throughout under growth factors influence. In this, post-response plays a very important role. Caries or trauma triggers an immune response. Balanced inflammation promotes healing, while chronic inflammation results in necrosis and periapical pathology. The microenvironment here is of very much importance, in which factors like oxygen tension, local pH and bioactive iron availability play a role, in which they will determine whether the pulp tissue progresses towards regeneration or irreversible tissue damage. So coming to the biomaterials that are used currently in the market, the MTA and the biodentin, these are the calcium silicate based cements inducing mineral deposition that form the dentine ridges. These are biocompatible in nature, though MTA has certain drawbacks of discoloration and handling while administration. Second comes the bioactive glasses. These release the calcium and the phosphate ion. It will precipitate into the hydroxyapatite formation. It will in turn promote regeneration and offer various antibacterial properties. The third comes the biomimetic restoratives in the form of adhesives and composites that are being engineered to mimic natural dentine through bonding and release protective ions release, and collagen linking conformers that improve the hybrid layer durability. Incorporation of nanotechnology also has come into play, in which there is a development of nanohydroxyapatite that restores early enamel lesions, and the silver nanoparticles and the zinc oxide provide sustained antibacterial and the microbial effects for targeted drug delivery in patients. The use of scaffolds in the form of hydrogel and 3D printed scaffolds have come into play that provide a structural framework for pulp regeneration by carrying stem cells and growth factors into the pulp chamber. Now coming to the MID that plays a role in diagnosis. So we have various modulations in it. First comes is the optical coherence tomography. This provides a real-time high imaging resolution of enamel and dentine without radiation. Examples are, we can detect the hidden proximal caries before cavitation, reducing unnecessary drilling and minimizing the aggressive dentine excavation and the enamel excavation. Also comes the use of fluorescence in the form of dipodent and QLF. It identifies early demineralization invisible to X-rays. The dipodent specifically detects the incipient occlusal caries lesion for prevention and intervention. And there have been advent of AI-based diagnostic radiographic analysis that help in detecting proximal caries lesions in the periapical pathology. Specifically, the example is Dental Mind AI highlighting suspicious radiolucencies on the bite-to-move radiographs that to avoid over-treatment. Second comes selective caries removal, which focuses on removing soft, infected dentine but preserving the firm, remineralized dentine. Stepwise excavation is done, so that is the partial removal of the caries in the deep regions, temporary restoration, and final excavation after pulp recovery. The deep molar lesion is managed by two-step excavation techniques such as with the use of calcium hydroxide liner, avoiding pulp exposure. Coming to the atraumatic restorative techniques, uses hand instruments and high viscosity glass ionica, specifically in the rural community programs, managing caries in children without electricity or drills. Coming to the non-restorative options, we can use the resin infiltration alcohol that is available in the market that penetrates porous enamel in early proximal caries and seam lesion. It is used in the white spot lesion on the maxillary incisor, post-orthodontics treated without drilling, and the casein phosphopeptide amorphous calcium phosphate CPP ACP are recently being used to replenish the calcium and the phosphate ions, remineralizing the early enamel lesion in the form of MI Paste Plus that is commercially available for remineralization of orthodontic white spot lesions. Also comes the bioactive peptides, specifically the P11-4 assembly. It is a self-assembling peptide scaffold that induces mineral deposition. The example of it is the Curodont applied to early smooth surface caries in young adults, specifically for the pediatric population and the adolescent groups. Third comes the pulp preservation through direct pulp capping, partial pulpotomy, and regenerative endodontics techniques. In direct pulp capping, we use the bioactive materials like MTA or biodentin where pulp is exposed due to trauma or caries. The example is traumatic pulp exposure in a young patient, permanent central incisor capped with biodentin, maintained vitality. Partial pulpotomy, removal of the inflamed coronal pulp tissue and sealing it with the various cements such as calcium silicate. Specifically for class 3 Ellis fracture in a 9-year-old managed with CIVEC pulpotomy and MTA use. Also in the regenerative endodontics, use of blood clot scaffolds or biomaterials have come into play that helps in regeneration process in the immature people, specifically in the pediatric and the adolescent group population. Examples are revascularization of a non-vital immature premolar using triple antibiotic paste and PRF scaffold. Also coming to the restorative techniques, what can be used as restorations? First is adhesive systems, second is the pit and fissure sealants, and third is the bioactive restorations in the form of GIC, and the recent advancement is the geomers. So coming to the adhesive systems, these allow minimal cavity decoration by bonding to enamel and dentine using universal adhesive and selective etchant mode for small occlusal composite restoration. Pit and fissure sealants prevent caries but progression in susceptible groups with non-invasive sealing. We can use the we have seen that high-risk 7-year-old receives resin-based sealants on permanent molars. Studies have been done on that. And bioactive restoratives such as the use of geomers release fluoride, calcium, and phosphate. The cervical caries are restored with geomers and for fluoride release and chemical adhesion. Now coming to the next part of it is integration of all these, that is the integration of biology, biomaterials and MID for the various restorative techniques and in active restorations, regenerative dentistry and others. So for active restorations, materials that can interact biologically, release ions into the to stimulate dentine formation, as I already told. Examples are geomers, which will continuously release fluoride, preventing secondary caries. In the use of regenerative dentistry, preserved tissue forms a basis for pulp regeneration using scaffolds and cells. In this, we have seen hydrogen scaffolds plus dentine pulpal stem cells in experimental models already that will regenerate the pulp dentine complex. Immune-informed materials, new cements induce inflammation and encourage healing, specifically through the M1 and M2 macrophage systems. that we will be discussing in the journal. Examples are chitosan modified adhesive demonstrate antibacterial and anti-inflammatory properties. The future vision of use of these materials as an integrative biology technique is that the smart biomaterials and AI-guided diagnosis can enable personalized dental care, pH sensitive smart composite release fluoride only in acidic conditions. These are being developed. So coming to the next part of it, what are the clinical and the research implications of these materials? First of all, in the clinical implications, vital pulp preservation materials like MDA and biodendine allow predictable outcomes in pulp caving and pulpotomies. They reduce the need for root canal treatment by maintaining vitality of the tooth and the natural immune defense of the tooth. Longevity of the restorations are preserved using bioactive restorations that will continuously release chloride, calcium and phosphate, reducing the onset of secondary caries and extending the restoration life. Adhesives with collagen use can be used to improve the hybrid layer stability between the layers and reduce degradation over time. Also comes the example of geomer restorations showing significantly lower caries recurrent rates rather than conventional resin composites in high-risk patients. The minimally invasive outcomes in MID approach conserve the natural tooth structure, improve aesthetics and reduce pulpal trauma. These approaches should align with the patient demand for conservative, less invasive and cost-effective care. Specifically, ICON stop progression of progressive dental lesions in adolescents without the need of drilling. Patient-centered benefit effects are reduced treatment complexity, results in fewer appointments and less discomfort, and patient cooperation. Vital retain the natural proprioception techniques, improving function. Examples are ART restorations in high viscosity GIC are in undeserved communities, specifically in the rural communities, provide durable fluoride release restorations without anesthesia or drilling. Now coming to the research implications, what can be done in future? There is a need for long-term clinical evidence. Many regenerative techniques are still based on in vitro or animal studies. Long-term randomized controlled clinical trials are required for proving their efficacy and safety. Examples are regenerative endodontics with P100 scaffold shows promise but lacks large-scale human trials because they have been done on mice models and rat models. They should be incorporated into rabbits and other mammals. Also, the cost and accessibility of advanced biomaterials is a concern. That is, a nanocomposites, tissue engineered scaffolds that are being developed are expensive, limiting use in developing countries only. Research should focus on making it affordable as scalable alternatives for community level ART programs, and standardization of the protocols need to be done. There is lack of uniform guidelines for MID techniques, pulp therapies and biomaterial use. Research is needed to define clear clinical pathways and modalities for different protocols in stepwise excavation of caries leading to variable outcomes across studies. What can be the future directions? Development of smart materials that respond to pH, bacterial and the microflora load, enzymatic activity need to be done, exploration of the stem cell based pulpal regeneration and translating it into 3D printed scaffolds for clinical translation, and the use of AI to guide diagnosis and optimize minimally invasive treatment plans need to be done. As I already informed that the Pearl AI and the Dialogcan can reduce over-treatment and standardize MID approaches globally. That needs to be done. So coming to the conclusion of it is that dentistry is transitioning from repair-based to biologically guided regenerative care. So by integrating the integrative biology, bioactive biomaterials that are being developed and minimally invasive approaches with ART, clinicians can preserve the pulp vitality, enhance the patient outcomes and move closer to the vision of true regeneration.